Welcome to this lecture on parasitology. Today, we're going to explore a fascinating and deadly microscopic organism known as Trypanosoma gambiense. This single-celled parasite is the primary cause of West African sleeping sickness, a disease that has profoundly impacted populations across sub-Saharan Africa. We will not only uncover how this parasite behaves and evades our immune system, but we will also use mathematical modeling to understand exactly how it multiplies in the bloodstream and how medical treatments can clear it. Let us dive into the microscopic world of this incredible pathogen. To begin our journey, let us establish exactly what Trypanosoma gambiense is. Biologically, it is a microscopic, single-celled parasitic protozoan that belongs to a specific class known as Kinetoplastia. Why is this specific organism so important for us to study? It is the primary causative agent of the chronic form of human African trypanosomiasis. You likely know this disease by its much more common name, West African sleeping sickness. This is a severe and fatal disease if left untreated, primarily affecting impoverished rural populations in sub-Saharan Africa, causing immense human suffering and economic hardship. Now that we know what the parasite is, we must understand why it is so incredibly dangerous. Imagine a highly skilled home invader who constantly changes their clothes so security cameras never recognize them. Trypanosoma gambiense does exactly this inside the human body. It is a master of disguise that evades our immune system through a fascinating process called antigenic variation. By constantly altering its surface proteins, it ensures that by the time your body's defense forces learn to recognize the invader, it has already changed its appearance and multiplied. This relentless cycle of evasion makes it a formidable pathogen. Understanding how this master of disguise enters the body brings us to its transmission cycle. This parasite is strictly transmitted through the painful bite of an infected tsetse fly, which acts as its biological vector. Once injected into the human host, it does not hide inside cells. Rather, it lives and multiplies extracellularly in the blood and the lymphatic system. As the infection progresses, the parasites eventually cross into the cerebrospinal fluid. This presence in the central nervous system leads to severe neurological symptoms, including the profound sleep disturbances that give sleeping sickness its common name. Before we move on to the mathematics of the infection, let us see this parasite with a concrete visual example. On your screen is a diagram showing Trypanosoma gambiense exactly as it appears in a patient's bloodstream. Notice the circular red blood cells scattered throughout. Among them, you can see the elongated, wavy forms of the parasites. The diagram highlights two critical anatomical features: the flagellum, which is the whip-like tail used for movement, and the undulating membrane that runs along its body. This extracellular lifestyle among the red blood cells is where the rapid multiplication takes place. Having seen where the parasite lives, we must now look at how rapidly it establishes a population. When the tsetse fly takes a blood meal, it injects only a small number of parasites. However, these organisms immediately begin to divide by binary fission. Because they are dividing continuously without immediate restriction from the delayed immune system, we can model this rapid multiplication using the mathematics of exponential growth. This mathematical modeling is crucial because it helps doctors predict the severity of the infection, known as parasitemia, allowing them to understand the threat level before symptoms even appear. This concept of rapid multiplication leads us directly to the exponential growth formula. The equation is P of t equals P0 times e to the power of r times t. Let us break down what each symbol means. P of t represents the total parasite population at a given time t. P0 stands for the initial number of parasites injected by the fly. The constant e is the base of the natural logarithm, approximately 2.718, which naturally describes continuous growth. Finally, r is the specific growth rate. If the growth rate r increases, the parasite population explodes much faster, overwhelming the host. Now that we have our formula, let us see this with a concrete example. Imagine a farmer is bitten and exactly 100 parasites are injected. The continuous growth rate is 0.5 per day, and we want to find the population after 4 days. First, we identify our variables and substitute them into the equation. We multiply the rate of 0.5 by the time of 4 days to get an exponent of 2. Evaluating e squared gives us approximately 7.389. Multiplying this by our initial 100 parasites gives a final answer of approximately 739 parasites. This shows how a barely detectable injection multiplies rapidly in just 4 days. So, we have proven that the parasite population grows exponentially. This brings us to the critical topic of medical intervention. When a patient is diagnosed, they receive medical treatment, such as the drug pentamidine, which actively kills the parasites. Because the medication removes the pathogen continuously over time, the population begins to shrink. Just as we modeled the rapid multiplication with exponential growth, we can model this continuous decrease using the mathematics of exponential decay. Understanding this decay process is absolutely essential for doctors to determine exactly how long a patient must be treated to ensure every last parasite is cleared. This medical intervention concept is mathematically represented by the exponential decay formula. The equation is P of t equals P0 times e to the power of negative k times t. This is nearly identical to our growth formula, but with one critical difference: the negative sign in the exponent. Here, P0 is the heavy parasite load before treatment begins. The variable k represents the continuous decay rate at which the drug kills the parasites. The negative sign mathematically guarantees that as time t increases, the total population, P of t, will shrink rather than grow, reflecting the effectiveness of the medication. Now that we have our decay formula, let us apply it to a clinical scenario. Suppose a patient has a heavy load of 10,000 parasites. They are given medication that clears the parasites at a rate of 0.2 per hour. We want to find the remaining population after 10 hours. We substitute our values into the decay equation. Multiplying negative 0.2 by 10 gives an exponent of negative 2. Evaluating e to the power of negative 2 gives approximately 0.1353. Multiplying this by 10,000 leaves us with a final answer of 1,353 parasites. This shows that even after 10 hours of effective treatment, a significant number of parasites still survive. We have covered a lot of ground today, so let us review the key takeaways. First, Trypanosoma gambiense is a highly adapted parasite that causes the devastating West African sleeping sickness, transmitted by the tsetse fly. Second, it is a master of disguise, using antigenic variation to evade the immune system while multiplying extracellularly in the blood. Third, we learned that this rapid multiplication can be accurately predicted using the exponential growth formula. Finally, we saw that medical treatments clear the infection following exponential decay, and our calculations proved exactly why patients must complete their entire course of medication to prevent a dangerous relapse. You now have a solid understanding of both the biology and the mathematics of this parasite.